Anyone have any questions before we get going? Okay, well, last time we talked about the structure of the heart, the anatomy of the heart. We went through the whole thing and we talked about the chambers, we talked about blood vessels, we talked about the location of the heart, we talked about, well, anyway, the whole anatomy of the heart. But in order to talk about how the heart works, first you have to look at it on a microscopic level. So we need to do a little bit of review of what it looks like if you look at it under a microscope. So back in AMP1, we saw this, and hopefully everybody remembers what it is. So remember the heart is these short cells. They're not long like skeletal muscle cells. They're short and they're branched. You can see how they're split apart here in different parts. So they're branched like this. They have only one nucleus. So let's see. For exams this semester, we're responsible only for the material covered in class or all the material from the PowerPoints and online reviews. Well, we are not covering lab material and the collaborates, but we are covering all of the material for the lectures. So if you're talking specifically about lecture, then yes, it'll be on these collaborates. And remember, I'm recording all of the sessions so that you can go back and review them if you want. Plus, not on Mondays and Wednesdays, but on Tuesdays and Thursdays and Fridays, I'm here to answer questions. But lab, we're not really covering that in the collaborates. But we have Anna's videos, which covers everything that you need to know. And also, she also has PowerPoints that are narrated. Now, if you don't want to do those, you don't have to do those. But definitely for lab, you need to go through and cover all that material on your own. If you have any questions, though, you can always bring them to class to collaborate, and I'll answer them for you. But anyway, let's go back to this. So this is that cardiac muscle tissue. And so, like I said before already, it's only one nucleus. These are short cells, they're divided. Remember, they're striated. You can see these stripes in here. And so in that way, they're exactly the same or very similar to skeletal muscle. And so I'm going to ask you in a minute to remember how skeletal muscle cells work, because we're going to see that's very similar. There is something very different and unique about cardiac muscle, though, and that's these things. Everybody remember what those are? What are they called? They're these. They're intercalated discs. And if you look at an intercalated disc, you blow it up. This is what they look like. Look, they're just channels is all they are. Now, if you look at it, the reason it looks dark is because the membrane of one cell is folded up like this, and the membrane of the adjacent cell is folded up like this. And so this dark area shows up right here where you have all these foldings. That's the intercalated disc. And here's what it looks like. Intercalated discs are just gap junctions. In other words, they're just channels. They're leak channels is what they are. So hopefully you remember leak channels back from AMP1. And what these are is there are sodium leak channels. So remember, sodium comes into a cell. Well, if you've got these leak channels, not only is it going to stay in this one cell, it's just going to zoom right on through to the adjacent cell. And so because of these leak channels, the cells are connected to each other. They're connected electrically. So if one cell goes through an action potential, because we have these leak channels in between the cells, if this cell goes through an action potential, then this cell is going to go through an action potential. If this one goes through an action potential, this one's going to, and then this one, and then this one. And so the cells are linked together. And it's because of these intercalated discs, they link them together electrically. And so if you look at these cells, they almost act like they're one giant cell. When you have one giant cell, remember it's called a syncytium. Skeletal muscle cells, remember, are a syncytium. But cardiac muscles are not a real syncytium. They just act like they are. So they're called a functional syncytium. In other words, they behave as if it's one giant cell. So the heart, then, because it behaves this way, acts as a unit. In other words, when one part of the cell contracts, when one part of the heart contracts, then eventually it's all going to contract because it's going to spread through the muscle cell. And so then this thing acts like a single unit. Does that make sense to everyone? Any questions about that? So what we're going to see is that the action potential spreads. It spreads through those intercalated discs. And so if we have 3 cells here, let's say there's 3 cells, we took them out of the heart, they're laying here like this in a petri dish or something. If this one goes through an action potential, it forces that one to go through an action potential. And that one forces that one to go through an action potential. Remember, it acts like a unit. It acts like this one big cell. That's why it's called a functional syncytium. And so look how many cells there are here. I don't know, and I can't count them. But if this one cell right here goes to an action potential, look, there's a intercalated disc. It's going to force that one to go through an action potential. And then this one's going to go through, and then this one, and this one, and this one, and this one. And so all of these cells, because they're all linked together, they're all going to go through action potentials. They're linked electrically. And again, it's because of these intercalated discs. Any questions about that? Well, if we take one of these cells and look at it, it looks like this. So all this is a diagram of one of these cells right here. So if we blow it up and look at it, look, it looks a lot like a skeletal muscle cell. Look, it's got sarcomeres in it. And it has I bands and A bands and all of that stuff. And so just like we learned back when we learned in skeletal muscle, we have I bands, A bands, H zone, M lines, Z lines. We have thick and thin filaments. And again, just like in skeletal muscle, these thick and thin filaments are going to slide past each other, and so we're going to get a contraction. We also have a sarcoplasmic reticulum. You can see here, that's that blue stuff. And just like in skeletal muscle, what it does is it stores calcium. Everybody remember that? And so what's going to happen when we get action potential is we're going to get this calcium coming out in here. And that calcium is going to bind to troponin, just like it did in skeletal muscle. Troponin is going to change the shape. It's going to pull tropomyosin off the binding sites, uncover the binding sites. And then we're going to get myosin and actin binding. None of that is any different. Any questions about that? Now, if you don't remember all that, you should go back and review. And you can pull those same PowerPoints up from A&P1, but review skeletal muscle contractions. It's really not any different. We did that whole set of steps. Everybody hopefully remembers those steps. I even had you put them in order for quizzes. But go back and review skeletal muscle. And it's going to be very, very, very similar to what happens with cardiac muscle. So another thing we talked about with skeletal muscle, we said that there are these properties that all muscles have. Remember, excitability means that you can get an action potential. And then the action potential causes contraction. We're going to get this shortening and tension production. Remember, you can stretch a muscle. Well, the way you stretch a heart muscle is to put more blood in the heart. And just like putting more water in a balloon or more air in a balloon, it stretches the wall. Same thing here. And then, remember, muscle, if you remove this tension, if you remove this stretch, it's going to go back to its original length. It's elastic. So hopefully everybody remembers those properties. All muscles exhibit these 4 properties. So, what is excitability? You can produce what? An action potential, exactly. And then, what is that action potential going to cause to happen? It's going to cause this to happen. So what is contractility? Yeah, it's contraction. It's when it shortens, and we're going to produce tension. So we're going to get tension production. And then again, you can stretch a muscle, you can pull on it, it gets longer. And so if we make more blood come into the heart, we're going to stretch the walls of the heart. But if we stop doing that and we remove this stretch, then the length of the heart muscle cells are going to go back to the original length. So all muscle cells, whether it's smooth, whether it's cardiac, whether it's skeletal, they all exhibit these 4 properties. But cardiac muscle has 2 new properties, 2 additional properties that skeletal muscle does not have. And these 2 properties, one of them is the heart muscle cell is automatic. It's automatic. In other words, you don't have to tell it to contract. You can cut the heart out. And there's a little video, if you're looking at the PowerPoints, of a turtle heart. And what the guy did is he took the turtle heart out of its body, and he's holding it in his hand. So it's just laying in his hand, and here's the heart. And the heart there all by itself sits there and beats. Doesn't have any nerves, it doesn't have any blood vessels, doesn't have anything, but it'll just keep beating until it eventually dies. Now, a turtle heart, that may take an hour or 2. It doesn't take that long in humans, but the same thing happens in humans. Humans' hearts are automatic. They contract automatically without any nerves. Does that make sense to everyone? So if we look at the heart, it has nerves which come down from the brain to the heart. There are nerves, but I can actually cut those nerves. I can actually cut the nerves and the heart will still beat. It does not need to be told to contract. It'll beat all by itself. It's automatic. And then another property that the heart has, not only is it automatic, But if I watch the beats, they're not irregular. They're very, very consistent over time. In other words, there's a built-in rhythm. So the heart is automatic and auto-rhythmic. And so if I look at the beat, if you watch it, you get a beat, and then you get a pause, and then you get a beat, and then you get the same pause, and then a beat, and then the same pause. It's very, very, very regular. And so it has this built-in automaticity and built-in rhythm. And here's that thing I was telling you about. I can't click on it and collaborate and show you, but if you're on, you can click on this and it'll go to a YouTube video of the turtle heart. And so you can watch it, and you can watch it beat by itself, and you can also see the rhythm that the heart has. And so look, here's what that rhythm looks like. So we get a beat, and then there's a pause, and then we get a beat, and then there's going to be a pause, and then we get a beat, and there's going to be a pause. And it's very, very regular because it takes the same amount of time to go from resting potential up to threshold potential. Look, it takes that much time. And not only does it take that much time this time, look, it took the exact same amount of time the next time, and the next time, and the next time. And so it has this rhythm built in. Any questions about that? Any questions? So there are 6 properties that cardiac muscle exhibits. These 4, like all muscles, and then these other 2 that the cardiac muscle exhibits. So 6 properties. And you should know what those properties are. Okay, and then we're going to get a contraction. So if you remember skeletal muscle starts with an action potential. So we're going to get an action potential. And here's the action potential. Well, look at the action potential. We know all about this. Remember, there's a stimulus that's required for skeletal muscle. That's a neurotransmitter. It's acetylcholine. And we're going to get a graded potential right here. We're going to open those chemical gated channels, and it's going to be a graded potential until we reach that point. And remember, what happens there is voltage gated sodium channels open. And so sodium then is going to rush in. So sodium is going to enter like crazy until we get up here to remember about plus 30. And then those voltage gated sodium channels close, and voltage gated potassium channels open. And as soon as they open, the potassium is going to rush out. And this graph's going to go down. Remember, it goes down to about minus 90 before we go back up again. Everybody remember all that? So when we look at it, there are 2 things that are going on. On this side, sodium is entering. And then that closes right there. And then on this side, potassium is leaving. So there are 2 events going on here. 2 events. Sodium enters, potassium leaves. Does it make sense to everyone? What happens on this side? Sodium enters. What happens on this side? Potassium leaves. We can see that, and it happens over and over again. So look, there's our action potential. So what happened on this side? Sodium enters. What happened on the other side here? What happened on this side? Potassium leaves. That makes sense? So two events are going on in the action potential of skeletal muscle. Now you can do a bunch of steps. Remember we went through like 10 steps, but really just two things are happening that have anything to do with this action potential. Sodium enters, potassium leaves. And then remember what's going to happen is this action potential is going to cause a muscle contraction. Remember, the action potential is going to cause the release of calcium. Calcium is going to go to troponin. Troponin is going to change shape. It's going to move. We're going to open up the binding sites. As soon as we start myosin and actin binding, then we're going to get tension production. And tension's going to go up. Remember, it goes up, and then we pump the calcium back out. So we take the calcium out, we start covering up the binding sites, and we get relaxation. So the action potential causes the muscle contraction. Everybody remember that? But when we look at skeletal muscle, look, the action potential is basically finished. It's done before the contraction ever even gets going. So remember what happens if we were to stimulate this muscle cell again? In other words, give it another action potential here somewhere, we would get another contraction. It would look like this. Instead of having one contraction, look, there's an action potential and we got a contraction. But before it came back down, we gave it another action potential and we're going to get another contraction. And before we give it another one. And so what we're going to get is these action potentials, as they speed up, we're going to get less and less time to relax. Look, there's hardly any time to relax until eventually there's zero time to relax. And so we get this smooth, sustained contraction. You may remember what that's called when you have the smooth, sustained contraction that just lasts and lasts until we stop stimulating it. What do you call that? It's called tetanus. Everybody remember that? So look, that's tetanus. Everybody clench your fist. Everybody do it. Clench your fist as hard as you can and hold it as long as you can. Look, when you do that, your hand is not twitching. Nothing is moving. It's just staying contracted. It's staying contracted until you get tired and then you stop, but it's going to stay contracted. That's tetanus. So when you hold yourself up straight, in other words, you stand up straight, all the muscles in your back and your neck and your legs, all those muscles are doing this. They're doing tetanus. Otherwise, you'd be twitching, jerking, and jumping all over your body. But you don't do that. You get this smooth, sustained contraction That's what tetanus is all about. And it's really, really good for skeletal muscle. But think about this. Do you think tetanus would be a good idea for the heart? In other words, what would happen is your heart would contract, and it would stay contracted, just like your fist. It would just squeeze constantly. Do you think that would be a good idea? Why not? The reason it wouldn't be a good idea is because the heart is a pump. And a pump has to do two things. It has to contract to push the blood out, but it has to relax in order to fill up for the next beat. So tetanus would be a really bad idea for the heart. So something has to happen to prevent this from happening. We do not want tetanus in the heart. And what happens is, remember, these are action potentials, and these action potentials are causing this tetanus. So what happens is the action potential is going to be different. But before we go on, does everybody understand what tetanus is? Why tetanus is a good thing for the skeletal muscle? And why it's a bad thing for the heart? Any questions about that? Well, here's what prevents tetanus from happening in the heart. If we look at skeletal muscle, remember the action potential. There it is. The action potential is basically done before the contraction even gets going. So if I give it another action potential, I get another contraction. And if I keep giving it action potentials really, really, really fast, remember it's going to look like this, and eventually it's going to look like that. We're going to get tetanus. That's because the action potential is over very, very quickly. It only takes about 2.5 or 3 milliseconds. In the heart, the action potential looks different. Here's what the action potential looks like in the heart. Look how long it is. Not this little short 2.5 milliseconds. This is about almost 300 milliseconds. In other words, it lasts about 100 times as long. And look at the contraction here. This action potential is over before the contraction gets going. But down here, look, the contraction is over really before the action potential is. So there's something different about the action potential of cardiac muscle. So remember skeletal muscle, how many events were there? How many? 2. Remember 2. What happens over here? Sodium does what? Sodium enters. And what was it over here? Potassium leaves. And because we only have these 2 events, look, it only lasts this long. It lasts about 2.5 or 3 milliseconds. But if you look at cardiac muscle, there are 3 events. 3, not 2, 3. This one's the same. So what do you think happens here? Just like skeletal. Sodium enters. And this one over here is the same also, just like skeletal muscle. What happened? Potassium leaves. But there's a third event that happens here in the middle. And that third event is that calcium enters. So there's 3 events. And because of those 3 events, it takes a lot longer for the action potential to be finished. And so instead of taking 2.5 milliseconds, it takes about 250 milliseconds, 100 times as long. So you tell me what's happening here. What's happening? Sodium enters. That's exactly right. And again, what happens over here? Potassium leaves. Exactly right. But the thing that takes the longest is this thing. This third event. What is this? Calcium enters. And because it takes so long, it takes 250 milliseconds, the action potential actually lasts longer than the contraction. Here's a contraction. And the contraction is basically over with before the action potential is over with. And so what that means is that we can't give it another stimulus until the action potential is done. Here it is. We can't do it here. No, 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 no. You can't give it another stimulus till you get over here. And look, the contraction is already over. And so there's no way for contraction to do this. There's no way for it to do that, or do this, or do this. So because of that, because of this calcium, you cannot tetanize the heart. You can't do it. Does it make sense to everyone? So, 3 events. So again, you figure out what these 3 events are. There's one here, there's one here, and there's one here. So, at 0, at 2, at 3, what are these 3 events? And then how long does this whole thing last? Not 2.5 milliseconds, but 250 milliseconds. Make sense? And so because of that, the action potential is so long, 250 milliseconds. Because of that, the contraction is finished before the action potential is. And so because it's finished before the action potential is, then you can't tetanize the heart. So again, 3 events. So think about that for a second. See if you have any questions. If you do, I'll answer them, sorry. So very long action potential, 250 milliseconds. And because we have this very long action potential, we have this very long refractory period. And so again, you cannot tetanize the heart. And again, if you want to see why that's important, just think about, again, clenching your fist and pretend that's the heart. If the heart were to do that, it would not be pumping any blood. It would just be staying contracted. And if the heart doesn't pump any blood, there's no blood delivered. And so none of your tissues, none of your cells would be getting oxygen or nutrients. So look how different skeletal muscle is versus cardiac muscle. And it all has to do with this action potential. Make sense? Any questions? Everybody still with me? So if you haven't already done it, think about why it's important that you cannot tetanize the heart. You'd get one great beat, but then you wouldn't have any more beats. And the heart, remember, is a pump, and so it must relax in order to fill up. So it looks like the action potential of the cardiac muscle is lower, and that's exactly true. We'll talk about what this resting potential, I think, is what you mean. It is different. The resting potential is different in skeletal versus cardiac. But we'll come to that a little bit later. And then, let's don't forget, we have these intercalated discs. And so when you have the intercalated disc, the action potential goes from cell to cell to cell to cell. And so it's going to spread over the whole heart. But the heart is big. How many cells do you think are in the heart? There's a ton of cells, a ton of them. And so by the time that it's spread from here to this cell, 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 by the time it's spread, it would take too long. It would take too long. And it would take so long that it would be out of refractory. And so what would happen is you would get contractions that are coming from all directions all time. I don't know if you can see my picture, but this, if you look at this, this is what the heart would look like. In other words, every little bit of the heart would begin to beat all individually because it would just take too long. And so what we need some sort of timing mechanism, and we need some sort of delivery mechanism to get the action potential where it needs to go as fast as it can go. And so what we have is we have sort of a superhighway system in the heart. This picture is blue, but it's like a superhighway. In other words, it delivers the action potential much, much, much faster than it would if we went from cell to cell to cell. It's called the conduction system of the heart. And again, it's like a superhighway. So think about the difference. Let's say you wanted to go from, I don't know, Irving to Dallas. Here's Dallas all the way over here. But instead of being able to get on a highway to go from Irving to Dallas, you had to go through every neighborhood. So you go and there's a stop sign, and you go and there's a stop sign, and you go and you turn and there's a stop sign, and there's a stop sign, and there's a stop sign. How long would it take you to get from Irving to Dallas? It would take forever, exactly. If you've ever tried to do that, it would be ridiculous. So instead of having to do that, we've built these highways in. So supposedly you never have to stop. There are no stoplights, there are no stop signs, and you can just zoom. Now, I don't know how fast that actually works, but in the heart, it does work. So we have this conduction system. It starts with something called the SA node, or the sinoatrial node. And the sinoatrial node is a little node of tissue, special tissue. And the reason it starts here is because this part of the heart is the fastest part of the heart. It depolarizes more often than any other part of the heart. And if you don't have any other influences, it does it about 100 times per minute. So about 100 times per minute, this thing's going to go through an action potential. And then remember, the action potentials are going to spread. And what they do is they spread across the surface of the atria, just like this. And then it's picked up by the AV node, or atrioventricular node. The atrioventricular node is another node of tissue. It's in the right atrium, right where it joins on to the right ventricle. So it's right there at the boundary between the atrium and the ventricle. And it actually is the second fastest part of the heart. So if you look at the AV node, instead of depolarizing 100 times a minute, it depolarizes about 40 times a minute. Another thing that happens here is there's a slight delay. When the action potential gets to the AV node, it delays the action potential. It delays it about 100 milliseconds. And what that does is it gives time for the atria to contract before it gets passed on down to the ventricles. Again, that happens at this AV node right here, or the AV node right here. And then from the AV node, look, it's connected to this like super highway. And so we go from the AV node to the AV bundle, which used to be called the bundle of His. His is a guy's name. So here is the AV bundle right here. And look, it crosses from the atria to the ventricles. Well, there's a band of connective tissue right here. That connective tissue, let's use a different color. It's right here. And the connective tissue does not transmit action potentials. So look, there's a bridge right there. Bridge is this. So this AV bundle acts like a bridge to pass the action potential from the atria down into the ventricles. And then from the AV bundle, we go to the bundle branches. There's two of these. Just like we've used right and left over and over again, We're going to use it again. Right bundle branch, left bundle branch. And you can see them right here. There they are. There's the left and there's the right bundle branches. And they go all the way down to the bottom of the heart. They go down to that point of the heart, the apex. And then there we go to the Purkinje fibers. This is also a guy's name. He's Swedish, so it's not G, it's Yi. So Purkinje fibers. And the Purkinje fibers then go back up this way, and they deliver the action potential to all of the individual heart muscle cells. So that action potential then spreads. So it starts here, spreads over the atria, it's picked up by this, the AV node. It's delayed here about 100 milliseconds, and then it's passed across the connective tissue by the AV bundle. The AV bundle goes down, and then it splits. So we get the bundle branches, and the bundle branches go down to the tip of the heart, and then finally the Purkinje fibers go back up and deliver the action potential to the individual heart muscle cells. Does it make sense? So, and here's what those Purkinje fibers look like. You might think they're nerves, but they're not nerves. They're specialized muscle cells. They're specialized muscle cells. So that's our little super highway right there. So we don't have to go from cell to cell to cell to cell like driving through a neighborhood. You get on the super highway and you just zoom. Does it make sense? So look at this. This shows you the pathway that the conduction takes. And so again, it starts with the SA node, goes across the atria to the AV node. There it gets delayed, the AV bundle, the bundle branches, and then the Purkinje fibers, and then the individual heart muscle cells. Any questions about it? So this is a pretty good summary slide. So when you want to look at it, when you want to talk about it or think about it, come back to this slide. And not only does it tell you what's happening, it also tells you a little bit about what's happening. And remember, here's where we're going to delay. We're going to pause. There's the AV node, and then what gets contracted, and then it goes down, and then back up to the ventricles again. Any questions about it? Well, look, we can follow the action potential. So this yellow part is actually the action potential. So again, it starts right there at the SA node. And then look, it spreads over the atria. And then it's picked up by the AV node, passed down the AV bundle, and then the Purkinje fibers. So if you look at how this is working, look, on the atria, it happens from the top down. But if you look at the ventricles, look, it zooms all the way to the bottom and then turns around and goes back up. So on the ventricles, it happens from the bottom up. And that's exactly the way the contraction is going to happen too. So this represents the action potential, but the contraction follows the action potential. So the contraction happens in exactly the same pattern. In the atria, it happens from the top down. But in the ventricles, look, it happens from the bottom up. And so if you think about how the heart beats, again, I don't know if you can see it over here, but if you look at my picture, it beats like this. So from the top down and then the bottom up. And so if you think about what's happening, blood is going to get pushed in the direction the contractions happen. And so when this happens from the top down, blood's going to get pushed this way into the ventricles. And then when this happens, blood is going to get pushed this way out of the ventricles. Does it make sense to everyone? So, atria contract from the top down. Ventricles contract from the bottom up. Well, remember, an action potential of a skeletal muscle looks like this, and then we get contraction. The action potential of the cardiac muscle, remember, looks like that. We got 3 events, and here's a contraction. So if we look at the contraction of cardiac muscle, it happens between depolarization and repolarization. It happens between the sodium entering and the potassium leaving. It happens here. This is where the contraction happens. And so look, if we can look at the action potential on the heart, we can look at depolarization and we can look at repolarization. Remember, this is depolarization, this is repolarization. And so the contraction is going to happen between these two. So if we look at this, that's depolarization. It's yellow, depolarization. So look, it's going to spread over the atria. And then repolarization is going to happen in the same exact pattern. So here comes repolarization. So look, between these two, right here, between depolarization and repolarization is where we get contraction. And then the exact same thing happens on the bottom. Remember, depolarization is going to go all the way down. Repolarization is going to do the exact same thing. So between depolarization and repolarization is when we're going to get contractions here. Make sense? So the yellow and the orange, that's electricity. That is not contraction. But the contraction happens because of the electricity. So here's where the atria contract, right in between those two. And here's where the ventricles contract, right in between those two. Any questions? So here's the pathway. Where does it all start? Remember, it starts with the SA node. It's going to spread over the atria. Then we're going to get to the AV node. Remember, there's about 100 millisecond delay here. There's going to be a slight delay. And then that's going to depolarize, and then it's going to go to the AV bundle. It's going to go all the way down to the bottom of the heart by these branches and these Purkinje fibers back up to the ventricle musculature. Any questions about it? So see if you can label and talk about what's happening with these things. So what is this structure? What is this structure? What is this structure? What are these structures? What is this? Where's the delay? All those things we just talked about. So what is number one? That's the SA node. And remember, the SA node is where it all starts. And the only reason it starts here is because this is the fastest. That's the only reason. And then number two, over the surface of the atria. And then number three is this, which is what? That's the AV node. Remember, the AV node has a delay built in, about 100 millisecond delay. And that gives us time to contract. And then we have this. That's the AV bundle. And then these two. And what are these? These are the bundle branches. And then finally, all of these that go this way and go back up, those are those Purkinje fibers. Make sense? Any questions? So look, this is the fastest part of the heart. And because it's the fastest, it sets the pace for the heart. In other words, it sets how fast the heart is going to beat. And because it sets the pace, it's called the pacemaker. So this SA node is the natural pacemaker of the heart. Well, what do you think would happen if we were to destroy this pacemaker? Look, I'm going to get rid of it. What do you think would happen? Would the heart stop beating? No, it wouldn't. You're right. What's going to happen is the second fastest part of the heart is going to take over. So the second fastest part of the heart, anybody remember what it is? Yeah, it's going to be the AV node. So now this is going to become the pacemaker of the heart because it's going to set the pace. But remember, it's only about 40 times a minute. And that for most people, that's just too slow. It's not enough to deliver blood. You can live with it, but you don't have a very good quality of life. You can't really walk or climb stairs or do anything like that. You just get too tired too quickly. And so usually what happens is if something does happen to the SA node, so it's either not working correctly or not working at all, what happens is they put in an artificial pacemaker. So an artificial pacemaker is actually a really simple device. It's basically just a battery. It's a battery that discharges every so often. So there's a wire that comes down here, and the wire sets off action potentials in the heart. Now it's also a little computer, so that the computer is going to decide when to send those little voltages, those little charges down. And a computer is smart enough that you can adjust it You can even have it sort of sometimes do self-adjusting. But no longer is the actual heart in charge. It's now this artificial pacemaker. Make sense? So David said, so by pacemaker, is it related to blood volume, ejection, or heart rate that causes fatigue? So we haven't kind of got to this yet, but when you think about it, when the heart pumps, it's pushing the blood out. And so in general anyway, the more beats that you have, the more blood that gets pushed out. And so if we slow down the heart too slowly, that means that there's fewer beats, which means there's less blood. And the less blood means there's less oxygen. And less oxygen and nutrients is what causes the tiredness and the fatigue. So yeah, they're interconnected. Now they're not completely 100%. We'll talk about more what happens with the heart and the heart rate and so on a little bit later on. But for now, that's a very good explanation. More beats, more blood, too slow, not enough blood, not enough oxygen, tiredness, fatigue, and so on. And then Kayla asked, is the actual pacemaker attached to the heart, or is it just hanging around it? Now, are we talking about the natural pacemaker, or are we talking about a artificial pacemaker? Because the natural pacemaker, this SA node, is part of the heart. So if I took the heart out, the pacemaker would come with it. But if you're talking about an artificial pacemaker, that's usually here, somewhere way away from the heart. Now there's a wire that goes to the heart, but the battery and all that is usually under the skin of the chest. So it's actually outside the chest wall. If you look at a person, you look at their chest, you'd see a little square, not necessarily square, but you'd see a little raised structure, and you could feel it with your hand. Does that answer your question? You wouldn't want to put it inside the chest cavity because remember, this is basically just a battery. What happens to batteries after a while? What happens? They run out of power, exactly. And if they run out of power, it's not going to work and everything. So we need it to be convenient in order to be able to change the battery or to recharge the battery. And that's usually why it's on the chest wall, outside the chest cavity. Makes sense? Batteries need to be replaced every so often. The battery here, they usually last about 25 years though. They're atomic batteries and they last a really long time. You don't need much of electricity to get this going. You only need a time, you don't even need, probably need like a 1,000th of a volt. So 1/1000th of a volt, or even less than that maybe. So you don't use it up very fast. And so they last for a long time. Okay, well, when we look at this, remember what we're looking at is electricity. It's electricity. When we look at this other picture, this is depolarization, remember? Repolarization. That's what's happening. It's electricity. It's a change in voltage. Well, we can actually measure this change in voltage, and when we do, we get something that looks like this. And that is called an ECG or an EKG. It's an electrocardiogram. And what the electrocardiogram is the electricity. It's the action potentials. Now, there's not one action potential, there's thousands of them. Every cell is going to go through an action potential. And so an ECG is a composite of all of those action potentials. The first guy to ever notice this was a guy named Einthoven, a German scientist. And Einthoven developed this thing where he could measure electricity. And he actually ran around putting electrodes on everything, on plants, on all kinds of things, on his dog. And what he found out was that if you look at a mammal, like a dog or a person, if you put the heart in sort of a triangle, in fact, it's called Einthoven's triangle, you get a very, very particular, definite picture. You can put these electrodes on the arms and the legs, or you can put them on the chest wall itself, as long as this heart is in sort of this triangle. And what he saw was this. Well, somehow this has to somehow work with this. This is the action potential. And so what he figured out was that these waves represented different things. Well, he wanted to be able to talk about these waves. He wanted to publish this. And so he decided to name the waves. So he just decided to use letters of the alphabet. He didn't want to start with A because he thought that he had really bad equipment. It was brand new, and there might be something over here that somebody would come along and find one day. So if he started with A, what would you call something in front of an A? So he just decided to start with P. So when you look at it, it's P, Q, R, S, and T. And it looks just like this. So here's the P, here's QRS, it's called the QRS complex, and then here's the T. And he saw this over and over and over again, PQRST, PQRST, over and over. And what he figured out was that this electricity happened every single beat. So this P, Q, R, S, and T happened with every single beat. Later on, what people figured out was that this P, Q, R, S, and T, again, has to do with action potential. And remember, the action potential has these three events. There's this, which is when the sodium comes in. There's this, which is the calcium comes in. There's this when the potassium leaves. Well, look, this is a big change in voltage. This is not, it's flat. This is a big change in voltage. So when you look at the EKG, the only parts you see are where the big change in voltage happens. So we only are going to see these two. So if we look at it, the P wave represents when the atria are depolarizing. So atrial depolarization. And you can't see it, but look, it comes on over here just like before. And then it's going to go this way. So here is atrial repolarization. The only thing is, at the exact same time that the atria are repolarizing, the ventricles are depolarizing. So here's the ventricle depolarizing. Look, it happens at the same time. And the ventricles are much bigger than the atria, and so they hide this. They hide what's going on with the atria, so we don't see it. And then we have this, which is the ventricles repolarizing. So when we look at the P, Q, R, S, and T, P represents atrial depolarization. QRS represents ventricular depolarization, and hidden inside it is atrial repolarization. And then the T wave represents ventricular repolarization. So one more time. P wave, atrial depolarization. QRS is ventricular depolarization. And then don't forget that hidden inside it is this, which is atrial repolarization. But you can't see that. It's hidden. And then the T wave is ventricular repolarization. Does that make sense to everyone? So, one more time. What is this? What's going on here? Atrial depolarization. That's exactly right. And then, what's going on here? Remember two things: ventricular depolarization, but hidden inside it is atrial repolarization. And then, what's going on here? Ventricular repolarization. Does it make sense? So, this ECG or EKG, you can use either one. Remember, we have these definite waves. And so again, P wave, depolarization of the atria, and the SA node. QRS, depolarization of the ventricles, and then hidden inside it, don't forget, hidden inside it is atrial repolarization. And then the T wave, ventricular repolarization. So again, three things happening. Any questions? Can I do it one more time? Sure. So let's go back for a second. So remember we said that when we look at this, remember we have depolarization and repolarization. Depolarization and repolarization. And if we look at the action potential of a cardiac muscle, remember it looks like this. Here's depolarization and here's repolarization. This is when voltage is going up to plus 30. So there's a big change in voltage, and so we can see it on a graph. This is flat. There's no change in voltage, and so it's invisible on the graph. You don't see it. But this one, remember, the voltage is going way down to about minus 90, and it's, again, a big change, and so we can see it. And so if we look at the atria, this would be the P, and this would be the QRS. Make sense? If we look at the ventricles, this would be QRS, and this would be the T wave. So look, for the atria, here's our P wave, and then there, right there, would be QRS. So P, QRS. But look, at the same time the atria are doing this, the ventricles are doing their thing too. So at QRS, the ventricles are depolarizing, and at the T wave, the ventricles are repolarizing. Does that help now? So look, even though this is happening here, happens right here, it's hidden by this much bigger thing. So when we look at the P wave is atrial depolarization. Repolarization happens with the QRS, but it's hidden. It's masked, it's hidden. And then QRS, ventricular depolarization. And then finally the T wave, ventricular repolarization. Anybody else have a question? So it's 3. Well, there are 4 waves. There's P, QRS, and T. So P wave just has to do with the atria, nothing to do with the ventricles. And then QRS just has to do with the ventricles, nothing to do with the atria, because this part, it's hidden. You can't see it. So P, atrial depolarization. QRS, ventricular depolarization. T, ventricular repolarization. Make sense now? Any questions? That's not all you can tell by looking at an EKG. We can measure a bunch of stuff on an EKG. So we can measure how tall the waves are, how high up they go. We can measure how much space is under the wave. So you can measure that. You can also measure how much time it takes to go between waves. So these are called intervals. So we can measure these intervals. And all these things get looked at when you have an ECG or an EKG and you go to the doctor and they do that. They don't just look at this, oh yeah, there's a P, a QRS, and a T. There's a lot of information that can be gotten from an ECG or an EKG, a lot of info. Sometimes the waves are upside down. Sometimes they're way taller than they're supposed to be. Sometimes they're almost flat and there's nothing looks like is going on. And so there's a bunch of information that can be gotten from that. And if you look at this ECG, look at these different colors, you can see where the action potential is as the ECG spreads. So look, there's our P wave. Look, the action potential spreading over the atria. And then there's QRS and T. It's all about ventricle. But each little part of it, even though you can't see it separately, has the different area of the heart that it's associated with. So look at the very, very beginning. Remember who the pacemaker is? So where's this action potential going to be happening? Right there. It's going to be there. And it spreads over the atria. As it spreads over the atria, look at this. And so on and so on and so on. Does it make sense? Any questions? Okay. So I can't show you an EKG on the video, but again, if you click this, it should take you to YouTube. And so you can see a little video about the ECG or EKG. Same thing with this one. This one's actually quite good. So if I were you, I would watch this one. But we're going to get the same thing. So look, if we do it on an oscilloscope or if we do it on a piece of paper that's moving, what we can get is this ECG that happens over and over and over again. And if you look at them, look, they all look identical. P, Q, R, S, T. 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 And if we count how many of these things happen in 1 minute, that's going to be the heart rate, because each one of these is 1 per beat. And so if we count it and it's happening 60 times a minute, then that's the heart rate. Now, I don't know what this one is. You have to know how fast it's moving. But let's just say it's 60 beats per minute. So that means that 60 times per minute we're going to have this P, Q, R, S, T, and then there's a pause. This is when the heart fills up. And then SA node starts again, and we're going to get P, Q, R, S, and T. This is when the heart is pumping the blood out. So if you look at it, look, it pumps, and then it fills up. It pumps, and then it fills up, and then it pumps, and then it fills up. It just happens over and over and over again. And remember, it looks like this. It's going to start with this SA node. And that's right there. That's where that's going to start. It's going to spread over the surface of the atria. And that's the P wave. So it looks like that. And then we're going to pick up with the AV node. The AV node happens right here. And then it's going to spread down. As it spreads down, remember this is depolarization, we're going to get QRS. And then we're going to get repolarization. So here's repolarization. Look, that's the beginning of repolarization. And so repolarization is this T wave. So hopefully you can follow through all through this. And as this happens, that's happening. As this is happening, that's happening, and so on all the way through. So this is another good sort of summary slide. So when you want to think about it or look at it, this is a slide to come back to so that you can see it all in once. Any questions about the ECG or EKG or conduction system or any of the stuff that we've just talked about? So look, here's a normal ECG. See if you can identify the different parts. So, what is this? Anybody know? That's a P wave. And then what is this? That's QRS. And then what is this? That's the T wave. And you should be able to do that all the way through. Look, P, Q, R, S, T. P, Q, R, S, T. P, Q, R, S, T. And look, it's exactly the same in every beat. It looks exactly the same. And again, if I count how many of these there are per minute, I would know the heart rate. So again, I don't know what this is, but an average heart rate is 75 beats per minute. So that's average across all people, all ages, everything. That's the average heart rate. And that means that this QRS, all that kind of stuff, this ECG is going to happen 75 times a minute. But when you look at it, it doesn't always look like this. This is a normal one. But look at this one. It's still normal looking. Look, there's P, QRS, and T. And then pause, P, QRS, and then T. But look how few times it's happening per minute. If we were to count them, look, this person's heart rate is 46 beats a minute. Now, for some people, that's normal. But for most people, less than 60 is considered abnormal. And there's a word for it. It's called bradycardia. Brady means slow. So this person's heart rate is slower than it should be. But then when you look down here, look, it's still, again, normal. There's P, QRST, P, QRST, P, QR. But look how often it's happening. So again, if I were to count how many of these there are in a minute, look, it's 136 beats per minute. And for most people, that, unless you're exercising, is too fast. And there's a word for that too. This word is tachycardia. And tachy means fast. And it's when you have a heart rate of above 100 at rest. So less than 60 at rest, bradycardia. Higher than 100 at rest, tachycardia. But let's look at this last one down here. Can you see the ECG? Can anybody see a P or a QRS or a T? No, there isn't any. That's because the heart is not pumping the way it's supposed to. When you have something like this, what it is like all the different parts of the heart are beating individually. It's not beating the way it's supposed to. Remember, it's supposed to be top down, bottom up, top down, bottom up. But this is happening like here, and then here, and then here, and then here, and then here. And if you look at it, I don't know if you can see my hands again, but if you look over at it, it's just like a bag full of worms. All of the different parts of the heart are beating in a different place. And if you look at that, that is not a very efficient pump. It's not going to be pumping any blood at all. The heart's just sitting there, and it's basically just wiggling. It looks like a bag full of worms. Well, when that happens, that means that it's no longer doing this P, Q, R, S, and T. In other words, the SA node is not in control anymore. It's not controlling the heart. The heart is not beating the way it's supposed to. Well, there's a name for this too. It's called fibrillation. So fibrillation, the heart, all the different parts of the heart are beating individually, and it's not pumping any blood. And so if your heart is doing this and it's not pumping any blood, unless you fix it pretty quickly, you're going to die because you're just not pumping any blood. You're not delivering any blood to your tissues. So the way they fix this is they shock the heart. They use fairly high voltage and they shock the heart. Now it can be done on the chest wall. So they use these paddles on the chest wall here and here, and they send a pretty high voltage. And what it does when you shock the heart is it stops the heart. In other words, all parts of the heart are depolarized at the exact same time, and the heart stops. And then hopefully the SA node will go back in control again. And so the SA node will go, and then we'll get QRS, and then we'll get T, and then we go back to a normal rhythm. But when you shock the heart, it does not start the heart. That's what most people think. It has something to do with starting the heart. It does not start the heart. It stops the heart. And it puts all of the different cells in the same place at the same time. So all the cells are depolarized at the exact same time. And what's going to happen is it's going to flatline and then hopefully go back to this again. Does that make sense to everyone? Any questions? Any questions about bradycardia or tachycardia or fibrillation? So let's look at these here. Daryl, yes, that's exactly what it does. The defibrillator does exactly that. So again, you can, think of it as on the chest wall, but it can also be inside the chest. And so if it's inside the chest, it's still going to do the exact same thing. It's monitoring the rhythm. And if this happens, it shocks the heart. And it hopefully polarizes all the different parts of the heart at the exact same time. Did that answer your question? So let's look at these. See if you can figure out what's going on with A, B, C, and D. So if we look at A, does it look normal, first of all? So can you see a P, Q, R, S, and T, and then over and over? Can you see that over and over? Yeah, this is normal. And that's what a normal ECG tracing looks like. So look, there's P, QRS, T, P, QRS, T. That's exactly what we would hope that it would look like. But let's look at this one. Look at B. First of all, is it normal? It's not normal. It's missing something. What is it missing? Yeah, it's missing the P. Where does the P wave start? Do you remember? What is the P wave? What's the P wave? The SA node. That's exactly right. So this person, their SA node is not working. Well, remember, if you mess up the SA node, the heart doesn't stop beating. Somebody else takes over. Who takes over if you get rid of the SA node? The AV node. That's exactly right. And so look, if we look at this, we don't have any P waves. Look, there's no P wave. We have Q, R, S, T. No P wave. Q, R, S, T. And so this is what's called the rhythm from the AV node, or sometimes it's called the junctional rhythm. But it's the AV node that's in charge now. And look how much slower this heart rate is than this one is. So in the same space, look, I count 1, 2, 3, 4, 5, 6, 7, 8 in that space. But here there's only 1, 2, 3, 4. So this person's heart rate is really just about half this person's heart rate, or about half normal. Do you remember what the depolarization rate for the SA node is? Remember, it's 100. But what is the depolarization rate for the AV node? It's 40. So look, this person's still alive. Their heart is still beating, but it beats very, very, very slowly. And it's usually so slow that they don't have a very good quality of life. And so this person would be a perfect candidate for an artificial pacemaker. Does that make sense to everyone? Let's look at this one over here. So first of all, look at it. Is it normal? No, it's not normal. Look what's happened here. Look, this one looks pretty normal. Look, there's a P, QRS, and T. But then look what happened. There was a P and nothing happened. And then we had a normal one, P, QRS, and T, and then we had a P and nothing happened. So in other words, what's happening here is that some P waves are getting through from the atria down to the ventricles, and we're getting a P, QRS, and a T. But some P waves aren't going anywhere. We get a P and then nothing. And then we get a P, QRS, and T, and then we get a P and then nothing. So the problem here, remember there's this AV bundle. And the AV bundle, remember, crosses this connective tissue right here. It acts like a bridge. What's happening here is that some are getting through. They're crossing the bridge. but some do not. This also has a name. This is called heart block. And there's 3 degrees of heart block. First, second, and third degree. This one is probably second degree because about half are getting through and half are not. Does that make sense to everyone? And again, this is another candidate for an artificial pacemaker. So you need to make sure that they all get through. And so they run the wires down here as well as up here. And then we have this last one. So again, we've seen this one before. This is not normal. There's no P, no Q, no R, and all that. This is fibrillation. And this is the one that we're going to use a defibrillator. So do we need to differentiate between? You don't really need to know. I'm not going to ask you that. But basically, it's just degrees of severity. So first degree, most P waves get through. So in other words, only very few times is there not going to be this happen. So you get normal, 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 and then one that's not normal. Second degree, it's more severe. About half the P waves get through. And then third degree is the most severe. In other words, most of them do not get through. In fact, none of them do, actually. So first, second, third degree is just degrees of severity. So here are those exact same things that we just talked about, and then you can look at them and hopefully you get an idea, and maybe you should be able to try to recognize these. But normal, there's our junctional rhythm where they're just our AV node. Here's this heart block. Like I said, this one's probably second degree. And then there's fibrillation. Here's another video for you to watch, and I would watch this one as well. Any questions? Well, the first quiz is over just anatomy. But we will have a quiz over what we covered today. So all of this stuff, ECG and SA node and all that kind of stuff, the action potential of the cardiac muscle versus regular skeletal muscle, all of that will be on the second quiz. But the first one is just from lab. It is just anatomy. It's also those blood vessels of the heart. Don't forget about blood vessels. But it's none of this stuff. It's just anatomy. The second one will be this stuff. I've not posted this one yet. Anybody else have a question? Well, that's all I have for today. So look over all this. You can come back tomorrow if you want, and I will do a review of this. We won't be covering new stuff tomorrow, though. But we'll review all this if you need to, and I'll answer any questions if you have any questions over this part. Any questions? Okay, well, thank you for coming, and thank you for your attention, and I'm glad you're keeping up with things, and if you have any questions when I'm not doing Collaborate, please email me, and I'll email you back as soon as I get the email. All right, so I'm going to stop recording now.